Prophase 1 starts off with the diploid cell. Unorganized chromatin lies within the cell as it condenses into the X-shaped chromosome. Through synapses, the chromosomes pair up with other chromosomes to form a tetrad. They begin to exchange chromatids with each other. This process is called crossover. Finally, the nuclear envelope disappears and centrioles move in to create spindle fibers. In metaphase 1, the homologous chromosomes line up at the equator of the cell and attach themselves to the spindle fibers. Moving on to anaphase 1, spindle fibers separate the homologous chromosomes toward opposite sides of the cell. The last stage of meiosis 1, the cell begins to divide with 2 chromosomes on each side of the cell. Spindle fibers disappear and the nuclear envelope reforms. Moving on into meiosis 2, in prophase 2, it is important to note that going into prophase 2, no DNA replication is done. Instead, both cells immediately form centrioles at opposite ends of the cells, which generates spindle fibers from them. Next, we have metaphase 2. In metaphase 2, the chromosomes align at the equator of the cell, where they then attach to the spindle fibers. Into anaphase 2, the chromatids are then separated by the spindle fibers and are led to the opposite ends of the cell. And lastly, in telophase 2, the spindle fibers once again disappear, giving way for the nuclear envelope to form within the cells once again. The cells separate through cytokinesis, the results of this whole process being 4 genetically different haploid daughter cells.